Good evening, students. This is day 3 on AC 23, the reverse transcriptase lab. So very quickly, just to go over what we did on days 1 and 2, originally what we did is we isolated some mRNA from mouse liver, and we used a enzyme called reverse transcriptase. And what it does is it takes mRNA and it turns it back into DNA. The trick there, of course, though, is that it doesn't have any of the introns that were originally in the DNA. All the mRNA is made up of exons. So what we have is a piece of what we call cDNA. It's not naturally occurring DNA like in the mouse, but instead a DNA that we synthesize that's completely made up of exons without any introns. So we made a whole bunch of copies of all of the different mRNAs that are found in the mouse liver, but that's not what we wanted. We were specifically trying to find the enzyme called GAPDH, which is down here. And GAPDH is useful in glucose metabolism. We picked GAPDH just because it's convenient. The mouse is always going to need to undergo glucose metabolism, and so we know that the GAPDH gene will probably be active most of the time because it will be producing this GAPDH enzyme. So GAPDH was chosen to study in this particular reaction just mostly out of convenience. Right. So then on day 2, what we did is we took all of the cDNA that we had, and we didn't want to amplify everything. All we wanted to actually amplify was our GAPDH section or gene. So we created some primers, and we put the primers just before the GAPDH gene, and then we added some Taq polymerase, and what happened is we did PCR, and PCR went through and it made a whole bunch of copies of just our GAPDH section. And we put that in the fridge until day 3, and so now on day 3 what we're going to do is we're going to run a gel to make sure that what we got is actually what we were expecting. Alright, so we're going to pour the gel just like we pour almost every other gel. We're going to use the Mini 1 gel electrophoresis apparatuses. You're going to take the gel out, take the comb out of the gel, place it in the carrier, pour 125 ml of buffer solution over it, and then you have to prepare your samples. So every tube that you get gets 12 ml of your master mix solution, which now has hopefully millions and millions of amplified copies of the GAPDH gene. And you're also going to add some loading dye. This is that purplish looking stuff, and all that does is keep it from floating out of the well. It adds some nice density to there. And so then we have 6 combs in our gel, right? So our gel looks something like this, and we have 6 combs there. All right, and all of these lanes are numbered, so 1, 2, 3, 4, 5, and 6. So we're going to have ladder, Group 1, Group 1, Group 2, Group 2 ladder. And the only reason that we put ladders on both ends is just to make it a little bit easier to read. Right? If you only have ladder in lane 1, the group who gets lane 5 is sometimes not easy to tell exactly where that ladder is. So you can run it on both sides just to make it a little easier on yourself. So once we load 10 microliters of each of these solutions into the wells, we're going to turn the gel electrophoresis apparatus on, and that will run for about 15 or 20 minutes or so. And what we should hopefully see, you can see down here that we put the ladder that we're going to use. Now we know from looking at the primers that were constructed that our DNA sections should run at about 500 base pairs or so. So that would be 0.5 kilobases. So if we have our ladder, you can see it's also the third bright line. So here's a really bright line, there's number 1. Here's the second bright line at 1 kilobase, and here's the third bright line at 0.5 kilobase. And so what we'd do is we would look down at our ladders, right, and we would get, I'll use red, we would get a really bright line, and then we would get a few more, and then we'd get a bright line. And then we get a few more, and finally we would get our third really bright line. And we would hope that this would be about 0.5 kilobases or so. And then hopefully what we do is we would see our little gene. I guess I could go sort of accurately colored here, right? A little green. We would hopefully see one band in all of our wells there. And so then, of course, the other ladder would run very similar on the other side. I don't need to draw this in, but just for the sake of completeness, I may as well. So here's red. I'll add another red end down there. Get some of these guys. All right. And that's what you're expecting to see. You're expecting to see some DNA glowing fragments that are about 0.5 kilobase pairs long. All right. I hope it works out for you.